lesson today is on diffusion. Constantly, molecules are moving in liquids, solids, and even gases. An example of this is when perfume is opened, the odor molecules of the perfume can travel throughout the room and fill it so that everyone in the room can smell it. A semi-permeable membrane can be used to model diffusion. You should start off by asking students if they know what a semi-permeable membrane is. And the answer to that is, a semi-permeable membrane controls what goes in and out of a cell by allowing only molecules under a certain size to pass through. So now we'll get started with our first demonstration. So first, break the students up into groups of 4 and pass out these bean containers. As you can see, here we have the larger beans. Then on this side of the wire mesh, there are smaller seeds. The wire mesh represents our semi-permeable membrane. Ask the students to shake their containers. And you'll be able to see that the small seeds are able to pass through the wire mesh, but the large beans cannot. This is the setup we're going to use today. Inside this dialysis tubing, there are glucose and starch solutions. The dialysis tubing acts like a semi-permeable membrane, and students are going to discover which of the 3 molecules, iodine, starch, and glucose, can pass through the dialysis tubing. On the students' observation sheets, they have a diagram similar to this one. You should tell them that S represents the starch molecules, G represents the glucose molecules, and I2 represents the iodine molecules outside of the dialysis tubing. Tell the students that they need to know how to prove what molecules have moved through the semi-permeable membrane. They need to know how to test for glucose and starch. Now we're going to test for glucose. Pass out to each pair of students a cup of water, a bottle of glucose, 2 glucose test strips, and a pair of tweezers. First, using the tweezers, pick up 1 glucose test strip, test the water, and we will do the same for the glucose solution. Make sure students lay down the strips in the correct rectangle. Next, we'll test for starch. Pass out another cup of water, a starch solution, and a bottle of iodine. Have students squirt a small amount of iodine into the water and squirt another amount into the starch solution. A purple or black color indicates that there is starch in the solution. And for the glucose test, a green test strip indicates that there is glucose. Now we'll look at the paper molecules. Starch is a large polymer and is made up of hundreds of glucose molecules all chained together. While glucose and iodine are smaller and simpler. In our bean model we looked at earlier, starch represents the larger bean, while glucose and iodine, being smaller molecules, represent the smaller seed. Ask students to predict how the molecules will move through the setup with the dialysis tubing. Hand out this testing setup to each pair of students on the red plate. Be sure to warn students not to touch the materials. We'll begin by testing the solution outside the tubing for glucose using the same glucose test strips we used before. Remind students that if it turns yellow, there's no glucose, and if it turns green, that indicates a presence of glucose in the solution. If students' test strips are not turning green right away, Make sure they wait a few minutes before testing with another glucose strip, because it may take some time for the strip to turn green. Returning to the bean model, you should ask students which bean or seed represents the glucose in our dialysis tubing demo. The student should say that the small seeds represent glucose because it was able to pass through the dialysis tubing, like the small seed can pass through the wire mesh. Next, we will test for starch. Have students pour the rest of their iodine into the cup. If the water outside the dialysis tubing turns black, that means the dialysis tubing that the students were using had a leak in it, and they should work with another pair of students. In our experiment, the solution inside the dialysis tubing turned black while the water remained orange. This means that there was starch trapped inside the dialysis tubing that could not move out, but the iodine was able to move in through the dialysis tubing to react with the starch. To wrap up, we should go over the result of the experiment with students. Glucose gave a positive result, meaning that it could pass through the semi-permeable membrane, or dialysis tubing. The iodine did not change color in water, but it made the solution in the dialysis tubing turn black. This means that there was starch trapped inside. Referencing the paper molecules, explain to students that the large starch polymer was unable to pass through our semi-permeable membrane, while our smaller molecules, iodine and glucose, could pass through the semi-permeable membrane. To clean up, collect the dialysis tubing in a bag and pour the contents of the water cups down the sink, but return all containers to the lab.